Hi, I am TJ Puls, a PhD student in the Weldon School of Biomedical Engineering. I first want to thank the International Foundation for Ethical Research for supporting my graduate studies with this fellowship. My work is focused on the design and validation of next-generation in vitro cancer models which recreate disease characteristics and behavior outside of the body. These models are designed to assist with the identification of new anti-cancer therapies while minimizing the use of animals. The models I am creating focus on tumor metastasis, a process where tumor cells spread throughout the body and ultimately lead to patient death. Of the many factors slowing progress in treating metastasis, one notable shortcoming is the lack of metastasis models that can be used for anti-cancer drug development. I have chosen to focus my research on pancreatic cancer because it is one of the most invasive and deadly cancer types. An important feature of pancreatic cancer is something called desmoplasia. This term describes the increased fibrous or connective tissue in and around a tumor. This connective tissue, known as the extracellular matrix, or ECM, is deposited by non-cancer cells called fibroblasts. The main protein that they lay down is fibrillar type 1 collagen. These two components, the fibroblasts and the fibrillar collagen, have been implicated in promoting metastasis but are largely missing from industry standard tumor models. Traditional in vitro tumor models involve growing cells on a 2D surface, such as a petri dish, or in cellular spheroids where the ECM is not accurately represented. To create 3D in vitro models of desmoplasia, I am using a unique self-assembly collagen polymer. Our novel 3D models involve embedding cancer cells within a 3D network that represents the ECM. This network is composed of fibrillar collagen very similar to what is found in our body's tissues. The unique collagen formulation developed in the Harbin lab is also standardized to increase reproducibility and offers much greater user customization than traditional 3D culture materials. The advanced models which I am developing in the lab involves recreating the interface between a tumor and the surrounding tissue to study tumor cell invasion, the first step of metastasis. To do this, spheres composed of fibrillar collagen and pancreatic tumor cells are created and embedded within a collagen matrix using a custom-designed fabrication platform. This tumor sphere model accurately recreates the tumor architecture and gives us control of properties both in and around the tumor, something not possible with the cellular spheroids I mentioned earlier. Using 3D imaging and analysis, we can then quantify the extent to which the cancer cells are invading in the surrounding tissue, giving us a quantitative measure that can be used for drug screening purposes. With this model, we can also systematically add components such as fibroblasts, immune cells, or vascular networks to truly recreate the tumor microenvironment. Overall, because of the user customization, the ability to quickly and reproducibly set up experiments, our models show great potential for translation to use in pharmaceutical industry. Early results from this work are promising, and we hope that one day these advanced in vitro models will reduce the use of animals in the drug development process. Ultimately, we hope that our work will lead to the discovery of new therapies and improved patient outcomes. Thanks for listening.